I'm Alex Singh, and my research project for 15400 will be on dealing with evolutionary models. So first off, my research mentor will be Dr. Karja, and she joined CMU faculty in January 2019, so she's relatively recent faculty, and a lot of her research is in the computational biology setting, so researching like evolutionary dynamics, basically how can we model populations changing over time, and then with specific computational aspects with machine learning and digital pathology, in addition to looking at like the human body and looking at the spatial structure of like human cells and like tumor architectures within humans. Now on top of that, most of her work revolves around simulating and modeling events over time to better understand evolutionary mechanisms, to better understand over some continuous time period, how does one population or one group change over time. So the main significance or importance of this is that models are vital for our understanding of how the real world actually works. First off, better models help us understand the real world. So although they are some abstraction and can't capture everything perfectly, we want to be able to get our models to be as close to the real world as possible. That way we can have some notion of like what's actually going on and be able to analyze these models and actually prove that this is the way things work. For example, models help us in conservation biology, where maybe we want to see, maybe some environmental change has happened, and we want to see how can we ensure that some species survives while their habitat is deteriorating. In a more clinical context, one type of application is uh, we want to eradicate pathogens or harmful populations of cells. So maybe if we give some environmental change, how can we expect like the pathogen or the harmful population or tumor to evolve over time, and how can we end up with this end goal of eradicating these cells? So this leads into stochastic modeling and how this is used within computational biology. So in computational biology, several types of models are used, and this will be more direct towards like my project, which will deal with like modeling tumor architectures. The first main model is evolutionary birth-death rate models, where essentially we can define basically at what rate we expect populations to be born and what rate we expect populations to die. And then we can combine this with things such as like Markov processes or a Monte Carlo simulation, where essentially we have some like distribution of like parameters and some like ranges of values that these parameters could take on. And essentially, basically, want to see is under some specific conditions, what does the population look like, or for whatever type of thing that we're modeling, what does that look like over time, and can we get some good insights from that? So one example is if there is a change in some environment, what is the likelihood that a population will survive over time? So in the image here on the right, what we see is we have some environmental change, and we have some individual initially with called like phenotype A, or that's just talking about the trait that they show. And maybe with this environmental change, we've either kept that phenotype in like the offspring with population with probability p, or we basically can redraw from like some new type of traits with probability 1 minus p. And we can model this over time and basically see as to like what kind of distributions the populations reach over time. So now more specifically into my project and the significance, that'll be on modeling like tumor environments. So initially, to start off with some background on how this works, a lot of models initially start off with some single cell, and basically this single cell may or may not have some properties that make it, very aggressive, like aggressive tumor that wants to like spread throughout the body. And we model this by essentially saying at each cell division, we create some new cells that may have some mutation that either makes it more prone to replicating quickly or like moving throughout the body. And with this model, we basically want to see how the tumor evolves over time. So in this image, A denotes the like first cell that we're starting off with. B denotes how we allow our cells to either like migrate or replicate. And then this image here shows basically when the new child cells are created, how that they can basically inherit some type of a mutation or proliferation from their parent, and then that can either mutate to give some different type of trait to the child. So ultimately, the main project goals are to better understand the mechanisms of trade-off between sort of migration, replication in tumor ecosystems. So what causes a tumor to migrate or metastasize, and what causes it to replicate, in its current place. And then this also leads into how does the spatial structure affect tumor populations in regards to the environment that it's in, what like the tumor looks like itself, and how it compares to like maybe it's locked in by other normal healthy cells. How do these various factors affect tumor populations and their growth? And then the very last question that kind of ties into it is can pleiotropy promote a sort of storage effect and lead to coexistence? Pleiotropy essentially just means you have like a single gene in your body that can produce like multiple different traits or outcomes. And so we want to see if this aspect, within tumors or within some other type of structure, can basically lead to having, different types of like tumor cells in the body exist at the same time. Maybe you have some tumor cells that are more prone to migrating. You have some tumor cells that are more prone to proliferating in place, and we want to be able to understand, what that looks like. And so ultimately, there's a lot of things that are unknown with this project, and the goal will be to like, get a better understanding through modeling and simulation of how these tumors actually evolve. Thank you.